The symposium, Sex Hormone Effects on Autonomic and Endothelial Function, was presented at the Experimental Biology meeting in April of 2015. The symposium was sponsored by the American Physiological Society, the Environmental and Exercise Physiology section, and supported by the Physiology Society. The goal of the symposium was to describe the impact of sex and sex hormone effects on blood pressure and vascular function, and to describe the mechanism involved in these effects. To accomplish this goal, we invited speakers involved in examining sex differences and sex hormone effects in human, animal, and molecular research. Dr. Michael Joyner opened the symposium by describing a series of studies conducted in his laboratories examining the role of sympathetic nervous system activity in the interaction between aging and sex in the regulation of blood pressure. In brief, while sympathetic nervous system activity does not predict blood pressure in young men or women, a strong relationship between SNSA and total peripheral resistance exists in young men, but not in young women. These findings are explained by sex differences in alpha and beta adrenergic activation between the sexes in young adults. Interestingly, in both men and women, aging strengthens the SNSA BP relationship, while interactions between autonomic and endothelial function, sex, and age become enormously complex. Dr. Joyner's symposium and paper serve to clarify these integrated systems. Following Dr. Joyner, Dr. Jeffrey Bender spoke on membrane-initiated rapid endothelial responses to estrogen. Dr. Bender, with Dr. Charlotte Usselman, expand this topic to describe the beneficial actions of estrogen on the vasculature in their review entitled Molecular Actions of Estrogen in the Regulation of Vascular Health, in which they describe both molecular mechanisms and functional outcomes. A major focus of Dr. Bender's talk was the discovery of plasma membrane-bound estrogen receptors, and that in humans, these membrane-bound receptors have been identified in endothelial cells, where the application of membrane-impermeant estrogens results in the rapid activation of eNOS, so are important for the regulation of vascular functions. It was these discoveries and other like them which opened new avenues of research into how estrogens might have their effects on the vasculature in women. The authors emphasized the importance of understanding tissue-specific signaling pathways in the success of hormone therapy. They described the benefits of selective estrogen receptor modulators, or SERMs, which have these tissue-specific effects and hold the potential key to improving cardiovascular health while avoiding negative effects in other tissues. Dr. Usselman moved the symposium back to human research with her talk, Sympathetic Baroreflex Sensitivity in Normotensive Pregnant Women. In her review, Effect of One Sex and Sex Hormones on Sympathetic Responses to Chemoreflex Activation, she addresses the question as to whether the chemoreflex might contribute to the cardioprotection afforded premenopausal women compared to age-matched men. The authors point out that few investigations have considered the effects of sex on sympathetic response to chemoreflex stimulation, with studies either excluding women or not considering sex as a variable. Generally, differences in the chemoreflex between the sexes are less apparent than differences due to sex hormones within women during the course of the menstrual cycle or while women are taking hormonal contraceptives, suggesting an interaction between sex hormone exposure and the chemoreflex in young women. Further changes occur during pregnancy and aging. Importantly, interactions between sex hormones and the chemoreflex have important implications for sleep apnea syndrome, a syndrome associated with a myriad of morbidities, including hypertension, endothelial function, and type 2 diabetes. Dr. Meredith Hay presented the final talk of the symposium. This review is immune system-related hypertension associated with ovarian hormone deficiency by Sandberg et al. Summarizes the role of ovarian hormones and sex in inflammation-related hypertension. Earlier work on the immune system interaction with blood pressure regulation had been primarily conducted in males. This review describes the potential role of T-cell profile in sex differences in hypertension in rodent models. Also of great interest, the authors describe the impact of the sex of the T-cell donors in angiotensin II-induced hypertension. The authors go on to demonstrate changes in T-cell expression of the aging female brain and how estrogen may impact regions of the brain involved in blood pressure regulation. Taken together, this review provides an overall picture of how the adaptive immune system is involved in angiotensin II blood pressure regulation and how sex and sex hormones influence this interaction. These 4 excellent talks and reviews have clearly accomplished the goal of the symposium, to provide the attendees and readers with an understanding of the complexity of the relationship between sex hormones, vascular function, blood pressure regulation, and cardiovascular disease. We address the relationship of estrogen to cardiovascular function in women from 4 perspectives: autonomic, chemoreflex, immune system, and molecular signaling. These excellent reviews demonstrate the importance of sex and sex hormones in many aspects of women's vascular function. These reviews indicate the complexity behind that most fundamental question regarding women's health. Why is estrogen protective against cardiovascular disease?